Okay, so we'll start again regarding the code translational insertion, which I was discussing in the earlier video. So this code translational insertion is going to happen in order to synthesize the membrane protein molecules, particularly the integral membrane protein, and in few cases even the eccentric proteins are also going to get synthesized by this particular process. So you can see in the first above figure, that is type 1 protein synthesis, what is happening there the initially it is going to be, the process is similar to the co-translational insertion, translocation process. So wherein first the polypeptide synthesis initiates within the cytoplasm, then through the SRP it gets stalled, and then it will be, whole machinery is going to get transported onto the rough endoplasmic reticulum onto a translocon. Then the translation is going to further restart with the removal of SRP and its receptor. Followed by that, there will be movement of polypeptide chain into the lumen of endoplasmic reticulum. So this is going to happen as usual. But during this particular process of translocation, in the third step, you can see there will be a patch of polypeptide chain, or I can say a sequence of amino acids which get synthesized, which have a very specific characteristics. So which make these particular amino acids, as soon as they get into the channel of the translocon, to get into an helical form or to get struck there itself. Why this happens? Because this particular sequence of amino acids are going to have specific set of information in the form of the arrangement of different amino acids, wherein here we find more of hydrophobic amino acids. What happens is that the channel internal environment is going to be more of hydrophilic in nature, whereas this particular part of the polypeptide chain is going to carry more of hydrophobic amino acids. So, and they are going to be present very close to each other or sometimes even in a stretch. So this makes this particular part of the polypeptide chain to make some kind of secondary structure by itself in order to protect their hydrophobic R groups. So these particular secondary structures, as they get created within the channel of this particular translocon, which makes the movement of this particular polypeptide chain further translocation into the lumen of endoplasmic reticulum difficult because of the size or steric constraints. Now this particular polypeptide, part of the polypeptide, why actually it is going to make itself like this? Because this is the signal which says this particular protein is a membrane protein and it is supposed to be transported or put into the membrane. So because of the pressure exerted by the polypeptide chain during its organization into some sort of secondary structure, which makes these translocon components, the Sex60O3 complex, 61 complex, and translocon complex component, this component to get open, and as it gets open, the adjacent hydrophobic environment is going to pull that particular part of the polypeptide chain which is have more of hydrophobic in nature. So because of the suitability in their environment, this particular part of the polypeptide chain, you can see in the fourth step, gets into the membrane. But when this particular part of polypeptide chain gets into the membrane, the translocation, that is, sorry, translational process is not going to be halted. It's still going to be continued on the translocan, but translocation is going to get stopped. And after the completion of the synthesis of polypeptide chain at sixth step, you can see the ribosomal complex gets off, leaving that particular polypeptide chain as it is in the membrane of the endoplasmic reticulum. So here in this case, this particular polypeptide chain or protein, the membrane protein, will have cytoplasmic side, it will have its C-terminal end, and at the lumen side it's going to have its N-terminal end. And during the particular protein's function, that is either when this particular protein is going to get transported and gets itself attached to a particular membrane, either into an argillary membrane or into the extra means plasma membrane, the portion of the polypeptide chain which remains within the luminal side will be the part of exoplasmic side, and the portion of the polypeptide chain which remains within the cytoplasm side will remain always as a cytoplasmic part of the polypeptide. Okay, so that is type 1 protein, how it gets synthesized. And the sequence, you can see the one which actually stops the movement of the polypeptide chain into the lumen of endoplasmic reticulum. That particular sequence is called as stopped transfer anchor sequence, because this will stop the translocation process, as well as it anchors this particular polypeptide chain. This portion is the anchor which makes it into move in the membrane. So that's why it's called a stop transfer anchor sequence. So it's a very specific one, a sequence which is actually also the signal sequence, which signalizes that this particular protein is a membrane protein and need to be inserted into the membrane. This is co-translational insertion process. Similarly, the type 2 protein molecule also going to get inserted into the membrane, but only thing is, in case of type 2 protein, there is one small change which we are going to see. Here the nascent polypeptide chain which is being synthesized will be initially getting translocated into the lumen of endoplasmic reticulum. But during its translocation, what happens is that there will be a series of positively charged amino acids are going to get synthesized. As soon as the series of positively charged amino acids get synthesized, the translocation of protein into the, so that is polypeptide chain, into the lumen of endoplasmic reticulum is going to get somewhat altered. The alteration is that a N-terminal portion which actually earlier hanged into the lumen of endoplasmic reticulum will start slowly moving outside. That is, this particular polypeptide chain now starts moving in a reverse way through the translocan and comes out of the translocan, and further synthesis of polypeptide chain by the ribosome leads to the synthesis of a sequence of nucleotides which is having a similarity with the stop transfer anchor sequence, which again stops the mobilization of this translocation, which actually, sorry, not stops the translocation, but instead just it is going to anchor itself into the channel. But the further continuation of the synthesis of polypeptide chain will get translocated into the lumen of endoplasmic reticulum. Now, here what we see is movement due to the movement of N-terminal side of the polypeptide chain into the cytoplasmic side, and further continuation of the synthesis of polypeptide chain leads to the dropping of the C-terminal end of the polypeptide chain into the lumen of endoplasmic reticulum. Before that, when this part of the signal sequence, which actually we call it as signal anchor sequence, gets synthesized, further synthesis of polypeptide chain and translocation puts a pressure on the translocan to get open. Once it gets open, the signal anchor sequence get sucked up into the hydrophobic lipid core due to the hydrophobicity interaction, and whereas the polypeptide chain which is being synthesized will remain within the channel of the translocan and get translocated further with the synthesis into the lumen of endoplasmic reticulum. And finally, once the translational process completes, the C-terminal will get dropped down into the lumen of the endoplasmic reticulum. So thereby, this particular polypeptide chain is going to have an orientation of N and C terminal opposite to that of first type of polypeptide chain. So wherein the cytoplasmic side now will have the N terminal side, and the C terminal is going to be the which is present at the lumen of endoplasmic reticulum. So this is the second type of protein which is going to get synthesized through co-translational insertion process. The other type, like type 3, if you come across with, which is actually nothing but similar to type 1 protein synthesis. Only the difference is that here, once the anchoring into the membrane takes place, then further polypeptide synthesis will not lead to the um, translocation, but still a large, means a lengthy chain is going to be synthesized at the C terminal end. In the type 4 type, so there will be a series of signal sequences are going to be present, which are going to have an alternative based on the orientation. If it is a normal orientation, then it is going to have stop transfer, signal anchors, followed by stop transfers, then signal anchor, like that. Or in the other way, it can have, you can see here, type 1 will have this kind of STA means stop transfer, SCA means signal anchor. So there is signal anchor sequence 1, which is going to have an opposite orientation, signal anchor 2, which is again having an orientation of N terminal into the lumen and larger C terminal into the cytoplasmic side. And there is type 4A, type 4B, which I was discussing earlier. So which is a multipass membrane protein, wherein type 4A, where orientation is such a way that N terminal will have cytoplasmic side and C terminal into the luminal side, which starts with the signal anchor sequence, that is type 1 sequence having positively charged amino acids at the N-terminal side, then followed by there is going to be a stop transfer, then SCA2, then STA, like that. In case of type 4b, which you can say some people will mention it as type 5, will have first a SCA2 type, that is signal anchor sequence, which is going to have positively charged amino acids at its C terminal side. Then there is again signal anchor sequence which is having positive charge amino acid at its N terminal side. Then STA, then SCA2, then STA, then SCA2, like that. So this will have now the orientation such a way that N terminal will be at the luminal side and C terminal is going to be at the cytoplasmic side. And there is one more kind of one which is not mentioned here, where both are going to be there at the cytoplasmic side or at the luminal side. So based on where you want this particular signal anchor and stop anchor and transfer anchor sequences of type 1, type 2, and they are going to be oriented properly, or they are going to be present in that particular fashion. So which is going to make the polypeptide chain to get inserted or stitched into the membrane into a specific way. So this is how the polypeptide chain synthesis is going to take place of type 1 to type 4. In some cases, we also have come across with some polypeptide chains which are going to be not an integral membrane protein, but they are going to be eccentric proteins, and sometimes even they can be the membrane associated proteins. That is the proteins which are not going to be present within the membrane by themselves completely, or they are going to be just associated, they just hang out of the membrane through some kind of anchor. They are going to be attached to the membrane. So here this is one example I'm quoting here, that is GPI anchored protein molecules. GPI stands for glycosyl phosphatidyl inositol. Glycosyl phosphatidyl inositol anchored protein. Similarly, there is farnesyl anchored proteins are there, geranyl anchored proteins are there. So varieties of anchored proteins we come across with within the membrane. Some are going to be always present, some are going to get anchored whenever it is necessary. So like varieties are there. So here in this particular case of GPI anchored proteins, this is one more method of protein which gets present within the membrane, gets incised into the membrane. So in this case what we see is, at the initial stage the polypeptide chain synthesis is going to take place properly, means in a normal way, wherein larger N terminal side it's going to have and smaller C terminal end it will have. Proper insert means it's a normal insertional process will synthesize this polypeptide chain. But once the insertional process is completed, later the protein like GPI transamidase enzyme is going to be playing an important role here. This transamidases, what they do is they are going to cut the peptide bond and transfer them onto another molecule carrying an amino acid and forms an amide bond. So that's why we call them as transamidases. So here GPI transamidase, what it does is, so it transfers the polypeptide chain, that is not completely, except the membrane portion, okay, at a specific position on this particular N-terminal part of the polypeptide chain will get cleaved and will get transferred onto the GPI, forming an GPI linked protein. Because this GPI is going to have a amine terminal, which is attached to the phosphate. That's why it is glycosyl phosphatidyl inositol. So this glycosyl phosphatidyl inositol portion will have this particular amine, to which the to that phosphoethanol amine, that amine is going to get attached with the protein by the amidases. This is another way the polypeptide chain are going to get attached to the membrane. Stop here. Continue in next class, next slides.